Hello students. In the last lecture, we studied about the introduction of coelom. Coelom as a basis of classification. Today, we will further see the functions of the coelom, types of peritoneum, and the origin of coelom. See, what, what are the significance, the uses, and the functions of coelom? Why it developed? A question can be asked that there are organisms which are acoelomates and then there are organisms which are coelomates. So what are the advantages coelomic animals have, coelomates have over the acoelomates? See, the first thing is the origin of coelom provides a new space called as the visceral space in which the new organs can be evolved, can be placed. Then coelom surrounds the internal organs, for example, the liver, intestine, kidneys. And what it does, the coelomic fluid protects them from the external shocks. Second thing, the coelom also serves as a hydrostatic skeleton. So, hydrostatic skeleton is any fluid when it acts as the skeleton is called as a hydrostatic skeleton. It can be water. In the case of coelom, it is the coelomic fluid. So we have seen porifera, sponges. Sponges, they also have hydrostatic skeleton, but that is not due to coelomic fluid. That is due to the presence of water. Similarly, Hydra. Hydra also has hydrostatic skeleton, but that hydrostatic skeleton is due to water, not due to coelom. So all those coelomic animals, organisms which have coelom, they have coelomic fluid, and that coelomic fluid acts as an hydrostatic skeleton. For example, we see annelids, earthworm. They have a hydrostatic skeleton. They do not have bones, cartilage. The skeleton, the body is given a specific shape, and this hydrostatic skeleton helps in locomotion and in maintaining the shape of body. Third, due to presence of the coelomic fluid, the coelom provides flexibility to the body. The internal organs in coelomate animals become large and are able to perform movements freely of their own. Fourth, in organism, it also helps in removing excretory waste. In some organisms, the excretory waste is released from the body into the released from the body into the coelomic fluid, and from the coelomic fluid, the excretory organs pick up the excretory waste and then they excrete it out from the body. Then it also helps in transportation of gases and the nutritive material from one part of the body to other. In some animals, it keeps the body wall moist to facilitate respiration and to destroy bacteria and other harmful microorganisms. And it also acts as a site for gamete maturation and the brooding of gametes. So these are some examples, some uses, function, and the significance of coelom. Such questions are asked. Give the significance or the uses or the function of coelom. And in another way, this question is also asked: what advantages coelomates have over acoelomates? That is, the organism which have coelom, how do they have any advantage over the organism which do not have a coelom? Now, which these things we have read earlier, but just to have a recap, because we will be needing this term again in this lecture. The term coelom was suggested by Haeckel in 1872. Coelom we had defined earlier, but we are just learning the definition again. The language is different, but the meaning is same. The coelom is defined as a fluid-filled perivisceral cavity lined by the epithelial cells derived from the embryonic mesoderm. And I have told you earlier also, these epithelial cells are which type? Cuboidal, columnar, squamous, which type? I told you peritoneum is an example of squamous epithelium. So these epithelial cells, they are squamous. The coelom is lined externally by a parietal epithelium or peritoneum. See, this is very important, the name. The external towards the body wall is parietal epithelium, and internally by visceral or splanchnic epithelium or peritoneum. Peritoneum provides support to the viscera and provides path to the blood vessel and lymph to travel to and fro. This is important. What's the function of peritoneum? It forms a cover first. Second, it provides support to the viscera. And third, it provides path to the blood vessels and lymph vessels to travel to and fro. See, what is parietal peritoneum? The parietal peritoneum lines the internal surface of the abdominal pelvic wall. It is derived from the somatic mesoderm in embryo. This is very important. The origin of parietal peritoneum is, it is from the somatic mesoderm of the embryo. These are the questions which are asked in competitive exams. Then it receives the same somatic nerve supply. Since it is derived from the somatic mesoderm, so it receives the same somatic nerve supply as in the region of the abdominal wall that it lines. Therefore, pain from parietal peritoneum is well localized. Parietal peritoneum is sensitive to pressure, pain, laceration, and temperature. So this is parietal peritoneum, which is lining the internal surface of the abdominopelvic wall. So what is the abdominopelvic wall? I'll show you in a diagram in the later slide. Visceral peritoneum. The visceral peritoneum invaginates to cover the majority of abdominal viscera. See, this is the important thing which we have to remember, that the majority of the abdominal viscera is covered by visceral peritoneum. It is derived from the splanchnic mesoderm. See, this is the difference. The origin of parietal peritoneum was from somatic mesoderm. The origin of visceral peritoneum is from splanchnic mesoderm in the embryo. The visceral peritoneum has the same autonomic nerve supply as the viscera it covers. Unlike the parietal peritoneum, pain from the visceral peritoneum is poorly localized, and the visceral peritoneum is only sensitive to stretch and chemical irritation. Pain from the visceral peritoneum is referred to areas of the skin, dermatomes. Now see, in this diagram, you can see this blue part which is lining the visceral organ. This is the liver. So this liver is surrounded by this blue peritoneum. Then this blue peritoneum again covers another organ. These are the part of intestine, another organ. So this blue part is covering the visceral organ. So that's why it is called as the visceral peritoneum. Now see, this is the abdominal wall. The abdominal wall is lined by this orange lining. And we read earlier that the peritoneum which lines the abdominopelvic cavity is the parietal peritoneum. So this orange lining is the parietal peritoneum. The blue lining which is surrounding the organ is visceral peritoneum. This diagram is taken from Teach Me in Academy. This is a copyrighted material which I am using here for study purposes. Now, if you see the cavity between this blue and the orange line is called as the peritoneal cavity, which we also called as the syllabic cavity. Now, see the organs, they are classified into two types: intraperitoneal organs and the retroperitoneal organ on the basis of the peritoneum which surrounds them. See, the definition of the intraperitoneal organs is these organs are enveloped by visceral peritoneum. These organs are enveloped by visceral peritoneum, which covers the organ both interiorly and posteriorly. Interiorly and posteriorly. See, this blue lining is the visceral peritoneum. This is a visceral organ in the center. This peritoneum comes and completely surrounds this organ. So you do not see any orange. The parietal peritoneum is not present. Only the blue visceral peritoneum is surrounding the organ. Such organ which are surrounded by completely by the visceral peritoneum, they are called as the intraperitoneum. Example is stomach, liver, spleen, jejunum, etc. Now this organ, if you see, is attached to the body wall by this double layer of visceral peritoneum. This double layer of visceral peritoneum is called as the mesentery. Now, this is the question which is asked in competitive exam. Mesentery is, they will give option double layer of the parietal peritoneum, double layer of the visceral peritoneum, single layer of the parietal peritoneum, single layer of visceral peritoneum. So we have to remember that mesentery connects the visceral organ to the body. So it is the visceral peritoneum which is double layered to form the mesentery. So the most of the organs, they are covered by mesentery and they are attached to the body wall. Then the retroperitoneal organ. See, the next type is the retroperitoneal organ. Now, if you see in this diagram, the retroperitoneal organ, the orange layer, this is the orange layer which is covering an organ. There is no blue visceral peritoneum covering. So this is the parietal peritoneum which covers the organ only from one side. The organ is not covered on both sides. So such organ which are covered by only parietal peritoneum, that too only on one side, are called as retroperitoneal. Now, if you see the question which is asked, which organs of the human body are not considered to be present as a part of coelom? Now, see, coelom is the yellow part. If you see in this diagram, this yellow part is the coelom. Now, if you see the retroperitoneal organ, it is outside the coelom. So, retroperitoneal organ, they are not directly present in the coelom. They are present outside the coelom because they are covered only by one type of coelom. So, retroperitoneal organs, if you see the definition, retroperitoneal organs are not associated with visceral peritoneum. They are only covered in parietal peritoneum, and that peritoneum only covers their anterior surface. They can further be subdivided into two groups based on their embryological development. Primary retroperitoneal, the organs developed and remain outside the peritoneal peritoneum. The esophagus, rectum, and kidney are all primary retroperitoneal. Secondary retroperitoneal organs were initially intraperitoneal, suspended by mesentery. Okay, so they were initially intraperitoneal. They were surrounded by visceral peritoneum. Through the course of embryogenesis, when the embryo was developing, they become retroperitoneal as the mesentery fused with the posterior abdominal wall. So the mesentery fused the posterior abdominal wall. Thus, in adults, only their anterior surface is covered with the peritoneum. Now, the examples of secondary retroperitoneal organs, they are asked in many competitive exams, so we should know. Again, this diagram is taken from Teach Anatomy. Examples of secondary retroperitoneal include the ascending and the descending colon. A useful mnemonic to help recalling which abdominal viscera are retroperitoneal is the SAD pucker. SAD pucker is used to remember which organs are retroperitoneal. SAD. S stands for suprarenal adrenal glands. A stands for aorta. Then duodenum. Then pancreas, ureters, colon, kidney, esophagus, and rectum. These are example of the retroperitoneal organ. That is, these organs are covered only by which type of peritoneum? Only parietal peritoneum. They are covered on both sides? No, they are covered only on one side. Are they considered a part of coelom? No, they are considered outside the coelom. And retroperitoneal organs are of two types, primary and secondary. Now see, the cavity, the coelomic cavity inside the human body is present. So you can see in this diagram, there are cavities present. First, if you see the topmost, this is the thoracic cavity, and this is the abdominal cavity. And then we have the abdominopelvic cavity. Now, if you see the thoracic cavity and the abdominal cavity, they are separated by this lining is the diaphragm. This is the question which is asked. Diaphragm separates, so it separates the thoracic cavity and the abdominal cavity. Origin of coelom. See, there are various views regarding the origin of coelom. Clark enlists four theories which have been given time to time. First is the Gonocial theory, second is the Nephrocene theory. Now see, these two theories, they are not much used now because they had some shortcomings. So the Gonocial theory and the Nephrocene theory, they were given initially, but the accepted theories are the Schizocylic theory and the Enterocylic theory. So we have accepted theories, total four theories, out of which the Gonocial and the Nephrocene theories, they are not the accepted one, they were rejected later, and the accepted one are the Schizocylic theory and the Enterocylic theory. Now if you see the Gonocial theory, this theory was first given by Berg in 1885. It regards the coelom as the cavity of an expanded gonad, and its origin is based on the common association between the gonads and the coelomic epithelium. Berg believed that coelom initially arose in a segmented condition by enlargement and cavitation of the gonads after the release of gametes. One of the main drawbacks of this theory that it closely links the origin of coelom with the origin of metameric segmentation, and hence it is difficult to account for the unsegmented coelomates. That how coelom arose in the unsegmented coelomate, if this theory is taken as truth. Thus, theory has no embryological support because gonads do not arise before the coelom. This is a very important thing why this theory was rejected, that gonads do not arise before the coelom. So this is an embryological support against the theory. Then the Nephrocene theory. This was proposed by Langston in 1874. According to this theory, the coelom originated as an expanded nephridium. This theory, however, was never taken seriously because protonephridia are not found in all organisms, and excretory organs are also not found in all organisms. So if we go through this theory which says that the coelom originated from the expanded nephridia of flat forms, then the organisms which do not have excretory organs like the echinoderms, they have coelom. So if coelom is arising from expanded nephridia, then the organism which do not have nephridia, they cannot have coelom. So this theory was rejected, the Gonocial theory, Nephrocene theory. So we have seen two theories. Gonocial theory rejected due to embryological absence of support, and Nephrocene theory again rejected because excretory organs are absent in some organism, but they still have coelom. Then the Schizocyl theory. This is the theory which is accepted and we follow it. And the Schizocyl theory says that coelom arises from the splitting. See, the word schizo means split. So the theory says schizo split seal. Coelom formed by splitting of the mesodermal plates is called a schizocylic coelom. And this type of coelom first arose in annelids. This is the question which we have to remember, that schizocylic coelom arose for the first time in Annelida. Then second question, which is normally asked in competitive exam, is that coelom evolved for the first time in. So coelom evolved for the first time also in Annelida, and in Annelids the type of coelom which was present was schizocylic coelom. And the next is the enterocylic theory, which see, the word enteron means cut. So entero, archentron, when you see the gastrula, embryonic gastrula, this part in between the embryo is called as archentron primitive gut. That is the part which will form the alimentary canal. See in the diagram, these blue cells, they are endodermal cells. The black cells surrounding the embryo, these are the ectodermal cell, and the yellow cells in between are the mesodermal cells. So the blue cells are endodermal, yellow cells are mesodermal, and the black cells are ectodermal. Anterocilic, see archentron will form the gut. So the anterocilic hypothesis says coelom arouses by out-pocketing of the archentron. So out-pocketing, these pouches are called as the enteric pouches. So when the enteric pouches are formed, they are formed by out-pushing of the archentron. Now see, this in the first diagram, the blue cells, these are the endodermal cells. In the second diagram, you see the mesodermal cells, they have completely covered the space between the ectoderm and endoderm. So the yellow cells are completely filling. Now in the third diagram, you see the pink part is the mesoderm. So the mesoderm is arising. Okay. The blue cells, that is the archentron, is out pushing into the mesodermal cells. So these pockets are called as the enteric pouches. So the enteric pouches, they keep on growing. If you see in the next diagram, the enteric pouches are growing. And finally, the enteric pouches push the mesoderm and a space is formed in between. This space, which is lined by mesoderm on both the sides, is called as the enterocolic coelom. So the coelom is formed by formation of enteric pouches. So the enterocolic hypothesis says that coelom arises by outpocketing of the archentron in the initial separation of mesoderm from the endoderm. In simple word, pouches arise from archentron, which fuse together to form a coelom, and its example are echinodermata, hemichordata, and chordata. So these are the theories which are related to formation of coelom. Thank you.